Professor Philip Johnson of the University of California at Berkeley invited a group of scientists and philosophers to a small beach town on the central coast of California. They came from major academic centers, including Cambridge, Munich, and the University of Chicago, to question an idea that had dominated science for 150 years.
I think Pajaro Dunes represented a turning point for many of us. Individually, we all had questions about evolutionary theory, but when we came together, each person brought something of their own to the table, and suddenly we all had a glimpse of a new way of looking at life that none of us had individually seen before.
I would have to say that this was an intense period of time in my life. It just seemed that there was something here much more intellectually satisfying than the view that I had held up until this time.
Looking back on it now, I think that gave me the motivation to actually look at the evidence and just see where I thought it pointed.
I realized that this was bigger than any one person or discipline, and this was the beginning of a community of scientists who were now willing to face the fundamental mystery of life's origin.
I sometimes wonder why anybody talks about anything else, because this is the most interesting topic there is. Where did we come from? How did we get here? What brought us into existence? What is our relationship to reality as a whole?
You look at the incredible diversity and complexity of life, and inevitably the question arises, what brought all this into existence? Was it simply chance and necessity, undirected natural forces? Or is there something else going on? Is there a purpose, a plan, a design due to an intelligent cause? I think that is the fundamental question.
The scientists who came to Pajaro Dunes set out to re-examine the mystery of life's origin. For each had significant doubts about widely held evolutionary ideas. Among them, biochemist Michael Behe questioned how natural processes could have assembled the intricate structures found within living cells. Dean Kenyon was an evolutionary biologist who no longer thought that chemistry alone could account for the origin of life on Earth. And Stephen Meyer, Paul Nelson, and William Dembski were seeking a new approach, one that could explain the origin of the genetic information encoded in living organisms. These scientists and philosophers began to formulate an alternative to the central theory of modern biology, a theory born in the mind of a British naturalist. His name was Charles Darwin. In 1831, Darwin, then 22 years old, set sail on a 5-year survey expedition for the British Empire. He journeyed from England on the HMS Beagle, traveling around the southern tip of South America, then north toward a chain of volcanic islands in the Pacific called the Galapagos. On this desolate archipelago, 600 miles off the western coast of Ecuador, Charles Darwin encountered an extraordinary array of birds, reptiles, and mammals, the likes of which he had never seen before. For more than a month, Darwin studied plant and animal life, took extensive notes, and collected specimens. Then he left, never to return. 25 years passed as he developed a theory about how the diverse forms of life on Earth had originated. In 1859, Darwin published a book titled On the Origin of Species. Its impact on science and ultimately all of Western culture was dramatic. Darwin argued that all life was the product of purely undirected natural forces, time, chance, and a process he called natural selection.
For 2,500 years before Darwin, most prominent scientists and philosophers, people such as Plato or Newton or Kepler, viewed the world as the product of some kind of design or plan. But a fundamental shift occurs with Darwin's idea of natural selection, and a real change in scientific philosophy is set in motion.
Darwin was not the first scientist to propose a theory of evolution, but he was the first to offer a plausible naturalistic mechanism that could produce biological change over long periods of time. To understand how natural selection works, consider the finch populations Darwin encountered on the Galapagos Islands.
13 species of finches inhabit the Galapagos Islands, and they vary subtly in terms of their body size and shape of the beak. Darwin returned to England with 9 different species of these birds.
According to contemporary Darwinian theory, differences in the sizes and shapes of the birds' beaks are the direct result of natural selection. One example often cited involves species of seed-eating finches. Following seasons of heavy rain, small soft seeds are plentiful throughout the islands. Birds with short beaks can easily gather food. However, during periods of drought, the only seeds available are encased in hard, tough shells that remain on the ground from the previous year. In these circumstances, only birds with longer, sharper beaks can crack the shells and eat the seeds.
Those birds with the longer beaks survive because they can reach the food source, whereas other birds cannot. That long beak, then, confers what biologists now call a functional advantage. The finches with smaller beaks, unfortunately, die out from starvation because they cannot reach that food source. If the drought conditions continue, the environment causes a change in the features of the finch population as a whole. Over time, the long beaks are passed on to succeeding generations because those beaks enable the birds to survive.
Natural selection was a powerful idea. Physical variations that proved advantageous would be inherited by succeeding generations. Through this process, populations would be altered, and over time, fundamentally different organisms would arise without any form of intelligent guidance.
Darwin wanted to explain everything in the history of life in terms of undesigned, unintelligent natural processes. And when he looked for an explanation, what he found was that a process he could observe in domestic populations also operates in the wild. Now, Darwin himself was very familiar with domestic breeding. He himself studied pigeon breeding. And he knew that for centuries, human breeders had been able to make dramatic changes in populations by selecting only certain individuals to breed. Darwin really suggested that this same process operates in the wild.
For Charles Darwin, natural selection explained the appearance of design without a designer. There was no longer any need to invoke an intelligent cause for the complexity of life. In effect, natural selection became a kind of designer substitute.
Today, Darwinism is generally assumed throughout science and the academic world. Yet, despite its wide acceptance, a growing number of scientists and scholars, including those who met at Pajaro Dunes, now challenge key aspects of Darwinian theory.
When we came together at Pajaro Dunes, we certainly didn't agree on everything, but we did share a real dissatisfaction with the mechanism of natural selection and the role that it was playing in biological explanation. Natural selection is a real process, and it works well for explaining certain limited kinds of variation. small-scale change. We have lots of examples of that, in fact. Where it doesn't work well is explaining what Darwin thought it could, namely the real complexity of life. We have a finch beak, and then you've got the finch itself. A minor change in the structure of the beak versus the origin of the organism itself. These are different scales of phenomena. These are different kinds of problems. And the important problem for biology is to understand where natural selection works and where it doesn't, and why there's a difference. Evidence is very powerful, and all of us have the sense that if we let that evidence speak for itself, that it would lead us in a very different direction, away from natural selection and towards a different conclusion about the origin and nature of life on Earth.
Natural selection acts only by taking advantage of slight successive variations. She can never take a great and sudden leap, but must advance by short and sure, though slow steps.
It's really interesting to notice that the more we know about life and the more we know about biology, the more problems Darwinism has and the more design becomes apparent.
Since 1988, Dr. Michael Behe has investigated complex biological systems that seem to defy explanation by natural selection.
For the longest time, I believed that Darwinian evolution explained what we saw in biology, not because I saw how it could actually explain it, but because I was told that it did explain it. In schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution, a Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before. And the arguments seemed pretty convincing. And at that point I started to get a bit angry, because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and since have decided that Darwinian processes are not the whole explanation for life.
Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
There are as many molecular machines in the human body as there are functions that the body has to do. So if you think about hearing, seeing, smelling, tasting, feeling, blood clotting, respiratory action, the immune response, all of those require a host of machines.
when we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
In speaking on the topic of scientific naturalism and evolution...
During the early 1990s, at a series of academic conferences, Behe first shared his doubts about the ability of natural selection to construct complex molecular machines. One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It's had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising. For the molecular motors that drive bacteria through liquid, each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force, it has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of the machines. It's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
In evolutionary terms, you have to be able to explain how you can build this system gradually when there's no function until you have all those parts in place.
The irreducible complexity of molecular machines poses a severe challenge to the power of natural selection. According to Darwin's theory, even very complex biological structures like an eye, an ear, or a heart can be built gradually over time in small incremental steps. Yet, as Darwin made clear, natural selection can only succeed if these random genetic changes provide some advantage to the evolving organism in its struggle for survival.
As I have attempted to show, it is not necessary to suppose that the modifications are all simultaneous if they were extremely slight and gradual. Natural selection is scrutinizing the slightest variations, rejecting those that are bad, preserving and adding up all that are good.
But could Darwin's small, favorable variations have produced a bacterial flagellum? Some scientists doubt the possibility.
How could something new, like a bacterioflagellar motor and all the components that go with it, how could it develop out of a population of bacteria that don't have that system? When each change, according to Darwin's theory, has to provide some kind of advantage.
Imagine such a scenario early in the Earth's history. An evolving bacterium somehow develops a tail, and perhaps even the pieces necessary to attach it to the cell wall. Yet without a complete motor assembly, this innovation would provide no advantage to the cell. Instead, the tail would lie immobile and useless, invisible to natural selection, which by definition can only favor changes that aid survival.
The logic of natural selection is very demanding. Unless the flagellum mechanism is completely assembled and actually works, natural selection simply cannot preserve it. It cannot be passed on to the next generation.
The important thing to realize about natural selection is it selects only for a functional advantage. In most cases, natural selection actually eliminates things that have no function or that have a function that harms the organism. So if you had a bacterium with a tail that didn't function as a flagellum, chances are natural selection would eliminate it. The only way you can select for a flagellum is if you have a flagellum that works, and that means you have to have all the pieces of the motor in place to begin with. So natural selection can't get you the bacterial flagellum. It can only work after the flagellum is there and operating.
In 1996, Michael Behe published a book titled Darwin's Black Box. In it, he argued that natural selection, Darwin's designer substitute, could not explain the origin of the bacterial flagellum or any other irreducibly complex biological system. Instead, Behe concluded that the integrated complexity of these systems pointed to intelligent design. Darwin's black box created immediate controversy. Over 75 publications, including some of the world's leading newspapers and scientific journals, reviewed the book. Some scientists praised Behe's work, while others dismissed it as unscientific and religiously motivated. Behe's critics also insisted that he had underestimated the power of natural selection. They argued that the flagellar motor could have been constructed from parts used to build simpler molecular machines, like this needle-nose cellular pump. If the components of the pump already existed, they could have been preserved by natural selection even before the bacterial motor arose. This theory is called co-option.
It's essentially saying that evolution or natural selection at some point was able to borrow components of one molecular machine and build a new machine with some of these components.
Scott Minnick has studied the flagellar motor for nearly 20 years. His research has led him to challenge the co-option argument.
With a bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually, you're going to have to account for the function of every single part as originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity, for its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction of a house, where workers follow a detailed blueprint and plan for assembly. The foundation of a house is poured before the walls are erected. Plumbing and electrical fixtures are installed prior to enclosing the walls of the structure. Windows must be hung before siding is applied, and shingles are attached only after plywood sheets are nailed to the rafters. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now. A rod is added, a ring is added, another rod is added, a U-joint's added. Once the U-joint's at a certain size and a certain degree of bend, about a quarter turn, that's shut off, and then you start adding components for the propeller. These are all made in a precise sequence, just like you would build a building.
To build a motor correctly requires a complex system of machines that coordinate the timing of the assembly instructions. But how could natural selection construct such a system?
The co-option argument doesn't explain this. You see, in order to construct that flagellar mechanism, or tens of thousands of other such mechanisms in the cell, you require other machines to regulate the assembly of these structures. And those machines themselves require machines for their assembly.
If even one of these pieces is missing or put in the wrong place, your motor isn't going to work. So this apparatus to assemble the flagellar motor is itself irreducibly complex. In fact, what we have here is irreducible complexity all the way down.
We know a lot about the bacterial flagellum. We still have a lot to learn, but we know a lot about it. And there's no explanation for how this complex molecular machine was ever produced by a Darwinian mechanism.
150 years ago, scientists did not know about irreducibly complex molecular machines. Yet Charles Darwin anticipated the difficulty that systems such as these could pose to his theory.
If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down.
There are really two big questions in biology. How do you get new living forms with new structures like wings and eyes from life that already exists? And secondly, how did life originate on Earth in the first place? Now, of course, we know that Darwin spent most of his life formulating an answer to the first of these two questions.
Charles Darwin compared the history of life on Earth to a great branching tree. The base of the tree represented the very first living cell, and the branches were new and more complex life forms that had evolved over time from the first primitive organism.
Darwin was trying to explain how the branches on the tree of life originated. He was trying to show how natural selection could have modified existing organisms to produce the great diversity of plant and animal life that fills the earth today. But when it came to the base of the tree, which represented the origin of the first life, the first living cell, Darwin had very little to say. In fact, in The Origin of Species, he didn't even address the question of how life might have originated from non-living matter.
The only glimpses we have of Darwin's opinions on the subject appear in a letter he wrote to a colleague named Joseph Hooker.
Regarding the first production of a living organism, if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat and electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present such matter would be instantly devoured. But this may not have been the case before living creatures were formed.
During the final years of his life, Darwin did little to develop his idea that a primitive cell might have emerged from simple chemicals in the primordial waters of the early Earth. But later in the 1920s and 30s, a Russian scientist named Alexander Oparin formulated a detailed theory about how this could have happened. It was called chemical evolution.
Oparin thought that he could explain the origin of the first life using Darwinian principles. He envisioned simple chemicals combining and recombining to form larger molecules, and then these larger molecules organizing themselves with the help of chance variations and natural selection into the first primitive living cell.
Over the next 3 decades, many scientists worked to develop and refine these ideas as they pondered the questions both Oparin and Darwin had raised. How could life have evolved from simple chemicals? One man thought he knew.
The problem of biological origins is, for a very long time, I would say, has been a real deep interest to me just because of the scale of the problem, the importance of it. Where did we come from? Why are we here? All that kind of question. Probed from the point of view of natural science.
During the late 1960s and throughout the 70s and early 80s, Dean Kenyon was one of the leading chemical evolutionary theorists in the world. And like others in his field, he was trying to explain how life on Earth began through a purely natural process.
In 1969, Kenyon co-authored an important book on the origin of life.
Gary Steinman and myself thought that if we were to pull together all of the lines of empirical evidence that had accumulated by the mid to late 60s in one continuous argument, we were very enthusiastic about the possibilities for explaining the origin of the main life-building elements.
Despite his optimism, Kenyon faced a significant problem. To explain how life began, he first had to account for the origin of the essential building blocks of every cell that has existed on Earth. Large, complex molecules called proteins.
Proteins have a wide range of function in the cell, everything from structural requirements in terms of scaffolding of the cell, the cytoskeleton, to enzymes, where they're actually processing molecules to harvest energy or to build components of the cell.
Proteins do pretty much all of the jobs inside of the cell, except for storing genetic information. That's left to the DNA, the RNA. But all the day-to-day jobs, cleaning up the cell, making energy, it's all proteins.
Kenyon knew that proteins would have been as important to the first life as they are to living cells today. He also recognized the complexity of their construction.
By the 1960s, scientists had determined that even simple cells are made of thousands of different types of proteins, and the function of these molecules derives from their highly complex three-dimensional shapes. The irregular shapes of some proteins allow them to catalyze or trigger chemical reactions because of the hand and glove fit that they have with other molecules in the cell.